weird, like, and then I saw her. Yeah, that's probably. She was on Facebook. An ad popped up for that company. Like, never heard of it before.
So I never used these.
All right, well, let's go ahead and get started. Thank you all for joining us. And most of you have already had a chance to hear from Dr. Rock Levinson. He did our introduction to this. We started this all off in the fall. And some of us, of course, worked with Rock here in physiology and know him from there. But I pulled up one of your old CDs, Rock, because I was curious on a couple things and discovered I didn't know you had actually gotten your PhD from Cambridge, England. That was interesting. So he's got a PhD ago. Yeah, a little while ago. Yeah, and he joined our faculty in 1979. I was very young those days, and he's been here ever since. And he retired a couple years ago, became a professor emeritus. However, you wouldn't know that he's retired because he's here so much, and it's been quite active and helped us with many things in maintaining equipment and giving us his professional scientific advice on things we need for the department in terms of shared equipment and things like that, and also continues to do research as well. And so, Rock, I'll turn it over to you, and thanks for doing this once again. Rock's got a wide variety of scientific interests, and actually this is one I wasn't even aware of until I saw the title.
Right. So that's one of the things about being retired is you don't have to worry so much about little things like funding and relevance and all of that kind of thing. So this is actually a project that's ongoing, and I got into it through friends, of course. These are people that I've known for a long time since they were graduate students, and the wife of this husband and wife team, she's a marine environmentalist, and like many people nowadays are very interested in the impact of global warming on the marine environment, particularly coral reefs, which we'll go over those kinds of things, but which are absolutely essential for marine ecology, and they're dying off at a very fast rate. So people want to know what the mechanism is. I mean, I think most of us are convinced that it's due to global warming, climate change. It's a sort of taboo word in some circles these days, but in any case, that's what scientists are really focusing on. Okay, so how, anybody here scuba diver? Anybody here scuba dives?
Right.
Yeah, right. Yeah. Snorkel? Some guys must snorkel. I think so. So yeah, that's the kind of the thing. If you go to sort of tropical climes, you see things like this, especially if you're a scuba diver, very colorful reefs and lots of fish and whatever. And these are just very packed, environments. There's a very complex ecology, with animals and algae and things like that. They're really very beautiful. However, as you all know, nowadays, and about 10 years ago, I started noticing this stuff too on some of my diving trips. You start seeing stuff like this, which is basically, instead of all that nice color, you have just stands of white color coral. It's called bleaching, okay? The coral loses its color. And furthermore, what happens is that it's not just simply it loses its color, but if it's bleached long enough, it'll actually die. So what you can see here is this beautiful blue-tipped coral. I don't know if any of you guys have actually seen this stuff. It's really nice. It's brown, has blue tips on it. Well, here it is. The same reef. It's dying. Just a few months after this, and then basically it's dead within a few months. Okay. So what we know about it is that once it bleaches, the coral is not dead, but if it doesn't recover, within maybe a few weeks or a month or something like that, it then can die, and then it's an irreversible kind of process. And what happens is that it gets overgrown with just algae. You can see the furry stuff and whatever, and it looks just really ugly. It's just not very interesting. Okay, so so there's stages. There's healthy. When it starts to become stressed like this, it becomes pale, and then it can become bleached and dead. Okay, so here's a particular example of beautiful blue coral in a in a reef surrounded by dying stuff and whatever. So it's really kind of tragic just from aesthetic point of view. However, you might ask, well, why should we care otherwise? So there, aside from the fact that we're all kind of immersed in biology here, biomedical science and things like that, it turns out that coral reefs really harbor, because it's a complex environment, about a quarter of the total species in the oceans. So they're home to that. And secondly, because they're the sort of the genesis of a lot of food sources for fish and the food chain and everything like that, a number of places in the world depend on them for food. So they're a very important food source. Here it says 500 million people, it's 500 million people. That's a substantial amount of the world's population depends on these things for a living. There's the tourism aspect that I think most of us have been part of, but there are actually more profound significance to coral reefs. First of all, the way that they've sort of evolved and everything like that, they grow in shallow water, and we'll go over the reason for that. And being in shallow water, they form a natural barrier to heavy waves and surf and things like that. So human sort of settlements and civilizations have often grown up in areas where they've been protected by these reefs. And now with increasing storms destroying reefs and the failure of reefs to regenerate, this is becoming a big problem. That's changing things. And then finally, there's this thing, which is just something very recent. Search for new medications based on natural substances has been going on for some time now, but it's mostly been concerned with sort of land things like plants, land-based plants or animals or microbes in the soil and whatever. But now people are beginning to realize that all the critters and organisms on reefs have evolved a number of mechanisms and things like that, which they produce compounds which may have very profound medical uses. And already anti-cancer agents and anti-inflammatory agents are being discovered. And they reckon that there are possibly about 10 times more compounds waiting to be discovered on reefs than there are, in forests, things like that. So this could be in the future a source for a lot of very important medicines and compounds important to human beings. Okay. Okay, so it's just a sort of an aerial photograph. Reefs are common, throughout the world. They go up to temperate waters, but this is a picture of the areas that we, if you are involved in or in tourism and whatever, you can kind of see that there are a lot of reefs in the Caribbean area and whatever, particularly in shallow water. The more light blue kind of areas are areas of shallow water where you find a lot of reefs. But every one of these coasts, the scale is a little bit too small here to see. We'll have fringing reefs and things like that. So I got into this when my collaborators were actually on the faculty of the University of Puerto Rico. And the University of Puerto Rico has several campuses and things, and they're very interested in ecology that affects Puerto Rico. And this is an aerial photograph of where we actually were doing our studies. It's a little town called Parguera on the southwest coast of Puerto Rico. Really charming little place. Just a little resort town for Puerto Ricans. Most people don't really know much about it, but anyway, you can see kind of up here, the actual town itself, and you can see that the area here is kind of shallow, and that's actually an area which has been designated a marine sanctuary for purposes of study by the University of Puerto Rico, and they conduct often yearly surveys of coral health and things like that to get an idea of how coral is surviving and whatever. There's still a fair bit of coral around there surviving, but you can definitely see, they say year to year, you can see it dying off more and more, bleaching and that kind of thing. Okay, so this is the species that we work on. The coral comes in a lot of different shapes and colors, as you all know. They're the kind of the spiny branch corals and things like that. This is a typical example of a stony coral. It's called Porites. It's the thing that we study, and it's a major component of reefs. It grows to huge sizes and huge extents and forms a lot of reefs and whatever. And what we want to talk about now, I want to go over for a lot, is to tell you a bit about coral biology. It's really fascinating. When I started this project, I didn't know much about it. I remember from my high school biology that corals were just kind of one step up from selenorates and sponges and stuff like that. But in fact, there's a huge amount of complexity in these little critters. It's pretty amazing. So I'd like to tell you a little bit about that. First of all, if you blow it up, what you see here is you see these little rosette things. So each one of these things is an individual organism called a polyp, and the little things sticking out there are tentacles that they use basically to feed. Microorganisms will swim through the water and whatever, and the tentacles have little stingers in them. And this just shows you a more magnified version of these tentacles and things like that. And they basically will sting these things and then shove them down their craw, their mouth, like that. Okay, so the actual sort of gross anatomy of these things is that they exist as a sort of a live polyp, but they surround themselves, very importantly, with a exoskeleton that's comprised of calcium carbonate. Okay, and reefs are almost pure calcium carbonate with a lot of these little guys on the surface. And as you probably know, calcium carbonate is limestone. It's a component for things like marble, other parts of things. Limestone's being used in a lot of things like lime and whatever. And so the way these things have been deposited over the millennia, these reefs just basically keep growing and depositing and stuff like that. You have these huge deposits of limestone and marble when it metamorphoses. And it's mostly due to, coral reef formation. There's, some that's derived from mollusks, the shells of mollusks and other critters, but mostly coral reefs, okay? So let's see, what else I want to tell you about these guys. I wanted to tell you, just if you look at that little bubble up here, that these are kind of simple body plans. They have just two cell layers. There's an outer layer that's called an ectoderm and an inner layer called an endoderm. And on the tentacle, where you see it right here, the ectoderm, the outer layer, has a little nematocysts. These are little harpoons that basically serve to sting or attach or immobilize prey. They often have toxins and things like that in them. So that it's on these things, but not in these things here. And then the inner layer has something called zooxanthellae, which we're going to be talking a fair bit about, because these things are really interesting critters, and most people, including myself, had no idea about the existence of these things and their importance to coral as an organism. Okay, but anyway, in this part of the coral polyp, there's got to be a mechanism for depositing calcium carbonate. These cells are specialized, and so we'll go through the genesis of this now. So what happens is that coral reproduction is pretty interesting. It can vary a bit, but the corals that we're going to be talking about basically are what they call hermaphroditic. In other words, the coral contains both eggs and sperm, and they can produce sort of asexually, although in fact they don't, because if they did that, there would be no sort of genetic kind of mixing. In Porites, what happens is that they'll produce either eggs or sperm, and the eggs sit in the inside of that polyp. Okay, that would be the mother, basically. Sperm just kind of are secreted and will basically, swim into that and fertilize the egg. And then the egg is, undergoes divisions and whatever, and finally forms something called a planula, which is a larva. And this larva then swims out of the polyp. It's free swimming, and it swims around for a time, maybe about a week or a few days or whatever, until it metamorphoses into something which we call a newly settled kind of form. It looks for a surface, and basically it might be another, usually another bit of coral or rock or something like that, and it will settle down, and then it becomes a polyp, and then it's got to secrete around it its skeleton to become an adult, okay? So there's a little bit more to it than that, though. Here's the lifestyle of it. First of all, these planulae, these larvae, are basically released from individual polyps in a big batch of coral. They swim around and they metamorphose, and then they become a young primary polyp. And then what happens is that the coral grows from that single thing sitting down by asexual budding. It's a really interesting kind of thing. Let me show you some of the forms here. This is a planula, a free swimming thing. It's about the size of maybe half of a grain of rice. They're tiny. I'll show you some pictures of them in a bit of free swimming. This thing will then metamorphose to this. This is a picture I took of some of the things that we got that had metamorphosed. And you can see it's kind of like a sort of a disc shape, looks like an alien kind of being or something like that. This is under fluorescence, and you can see that at the mouth part of it, this is where the mouth is, there's a fluorescent protein that these things basically produce called green fluorescent protein. I don't know whether other speakers have talked about GFP, they call it. It's now in use as a kind of a tool to allow us to visualize a lot of other things in the laboratory. But it was first discovered in jellyfish, but it turns out that these coral guys have it too. Why? I don't think anybody knows what the purpose of it is, but they're kind of interesting. Anyway, if you look at this thing here, you can see these kinds of buddy structures. Those are the tentacles starting to develop, all right? Okay, let's see. And then finally, when these things settle down, after just a fairly short time, they basically become mature kind of polyps. You see these tentacles waving out, and they've secreted the skeleton, kind of around them. So one of the cool things about coral is the way that it actually grows. Okay, so you have a single polyp sitting there. What happens? What happens actually is that the single polyp will actually bud off and buds a new polyp, and this polyp basically will then sort of migrate to a different spot and form its own exoskeleton and what have you. Okay, so what happens is that you get these kind of colonies of coral, but the interesting thing about them is that they all remain connected together. So in other words, you can consider these things one organism. So that big chunk of parietes that I showed you, that's one critter. It's all connected. It's all the same kind of thing. So the other example of this kind of thing that we have here in Colorado, that's aspen. Aspen grows all, tend to be from the same tree. They have runners that run along and then put new aspen up. So some people say aspen grows are the biggest living organism in the world. There's some people who say that, coral reefs are the biggest single living organisms in the world in terms of mass, that kind of thing. But it's a really amazing kind of thing. So reproduction then takes place sexually. The planula larvae, these things just kind of swim out and then settle. And then when they settle, they bud and then, increase in size. And that's a very lucky thing because it means that you can grow these coral in the laboratory, and people are doing that, and they're repopulating the dead reefs now with these things, very successfully. And they grow fairly quickly. So here's an example of just a small budding thing of a few of these things. It takes maybe, I don't know, maybe a month or so for these things to do that. There might be one guy, that dude in the center might be the original one, and these are all the buds and whatever. And, eventually they form that and bigger things too. I don't know if you've ever been diving or anything like that, there's some brain corals I've been, these things are just enormous, they're just huge.
Can you give us a frame of reference for size?
Yeah, right. Sorry. Yeah. So this thing here would be probably a couple feet across. Yeah, but they can be much bigger too, or smaller, or whatever. In this particular area, you can see these things, these mounds. Each one of these coral mounds that you see in the background would probably be about that big. So this particular part of the reef in Pargera is not, a very mature, extensive, kind of reef that you find in some other areas. But it has a lot of diversity. It's nice. Still nice anyway. Okay, so here's another part that I wanted to stress about things, which is the whole thing about coral color. Okay. So this admittedly is kind of a little bit over the top, color enhanced kind of image. Things don't quite look like this when you're down. I mean, as you know, when you scuba dive, what happens is that as soon as you get down maybe 20, 30 feet, you start losing the red and orange colors. Things start looking blue. So then you get to see that if you bring along a light or something like that with you or whatever. But in any case, there is a lot of intrinsic color to coral. It's pretty amazing, especially if you dive in an area. The place that I started diving was in Fiji, where the water was crystal clear, and down to about 30 feet, it was just all of these incredible colors. And, not quite that garish, but almost, really amazing. So the one thing that surprised the hell out of me when I started doing this is the knowledge that, in fact, coral themselves the coral polyp has no color at all. What happens during bleaching is that it has little algae that live within the cells of its endodermal layer. And these things are the algae, because they photosynthesize, they have pigments, photopigments, that are all unique, that have different colors and things like that. And the bleaching process is when the coral kick these things out. They basically, for some reason, when the temperature goes up, they don't like these things there and they leave. And so the coral that's left behind is just basically transparent, has no particular color. So yeah, let's go over some of the features of these zooxanthellae. So they're single cell algae organisms. They reside actually in the polyp tissue, and I'll show you a couple pictures just in a bit about what those things look like. So they're, don't worry about the word hermatypic. It means just, sort of like solid, stony reef-forming corals. That's a feature of coral organisms that do that. Not all corals have these, but most of them. Most of the solid corals do, the soft corals don't. They are very essential to helping deposit calcium carbonate, okay? I think how they're essential is not exactly clear, but has to do more with this thing here, is they provide nourishment for polyps. So these guys, a lot of them live in the coral, and they grab sunlight, and they photosynthesize, and they produce sugar, glucose, the energy source, the main energy source for animals, right? And so that's what they do for the coral. And it turns out that probably more than half of the energy that these corals use for reproduction, for reef building, which is just, big energy drain, is derived ultimately from these zooxanthellae, not from the coral themselves, okay? So it's a very important part of this sort of relationship. So it's symbiotic, meaning that there's something in it for everybody. And so what's in it for the zooxanthellae? Well, what's in it for the zooxanthellae is that the coral critters, they eat animal products and things, and they metabolize. They produce nitrogenous compounds that plant algae type things really need for their own metabolism. So both critters live. The thing that's really interesting about this, though, is that these guys actually live within the cells of the algae. So the symbiotic relationship is different. Let me show you a few things. These are pictures that I took of various things here. So you can, when you shine a light on these things, the zooxanthellae will fluoresce. And so these red, orange things here. Those are individual zooxanthellae. And this is one of those things looking down on the top of a larva that's just about to settle. It's gone from that planula swimming stage to the newly settled stage, and it's just about to settle. And the green color here is actually newly secreted calcium carbonate. So it's starting to make its exoskeleton at this point. Here's a free swimming version of it. Again, it's just about ready to settle down. You can see, again, around the mouth, that's the green fluorescent protein. You see these little buds here. Those are the tentacles starting to come out. Here's one which is even more developed. The tentacles are now starting to protrude. This thing is getting a base shape. That's the mature shape. And just very soon it'll be a sort of an adult kind of form, secreting its own skeleton and things. So this is something I did here. I have a confocal microscope which is able to optically section. It's just kind of like you actually made a section through this thing. You can see that there are just tons of these zooxanthellae within this critter. Here's the mouth area and whatever. And the question is where exactly are these guys? And this shows you a section in a planula, free swimming larva. And you can see that it's not in the outer cell layer. This outer thing here, that's the ectoderm. It's within the endodermal layer that these guys reside. And they're actually living right within those cells, okay? So it's different from the way that we normally think of symbiosis. I mean, the typical examples are things like a remora, little fish that kind of clean sharks. They kind of attach themselves to sharks. That's a symbiotic relationship. Or the cattle egret that sits on the back of water buffalo that eats parasites, helps the water buffalo, and of course provides a food source for the cattle egret, right? The cool thing about these things is that they live actually within the cells. They're actually integrated. They infect this thing. So from the very earliest time, when this planula develops, these things are apparently present. And it's not clear why it is that when temperature rises, when the coral gets stressed, why they kick these guys out. One of the ideas is that these guys are photosynthesizing. And as temperature rises, what happens is the metabolic activity of the zooxanthellae, the algae, goes up quite a bit. And when that happens, they not only produce a lot of good stuff like sugars and things, but they produce a lot of bad things like oxygen-free radicals. And you've probably heard about these things in humans. And, they're bad for you. They cause tissue damage and things. And so people think that there's some kind of mechanism that the coral polyp has to force these guys to leave. Right? What the mechanism is for showing them the door, nobody really knows. They just kind of leave, right? Once they leave, though, they will come back if conditions kind of improve and whatever. But it's not clear. The other thing to know is that, as you kind of can see from the that other picture that I had, that there are many different colors, right? So it means that there are many different kinds of zooxanthellae. They're called clades. They're kind of like subspecies of zooxanthellae. So, and they're all specific for just a particular kind of coral and their particular kind of conditions and things like that. So one of the things a number of us are thinking of is whether what we can do is re-engineer zooxanthellae so that they're more adapted to elevated temperatures and they won't, sort of anger the coral host as much or whatever. So there's a lot of work being done on that.
Yeah. So the mechanism behind what's affecting the zooxanthellae is temperature change in oceans. Is it also going to be the increase in acidity that's also associated with carbon?
Thanks. That's right. So, what Rob is saying is temperature isn't the only thing that's changing in oceans. There are other things, pollutants and particularly acidity. Yeah, and so there are some folks who are trying to determine what the effect of, say, acidity, which is a general thing, which is definitely increasing as well. Okay, and so that'll get us into our last thing, which is how does acidity increase in the oceans? Well, that increases because what's happening is CO2 is rising in the atmosphere because of burning of fossil fuels and things like that. And in the atmosphere, CO2 is soluble in water, it will dissolve in the ocean, and as we'll see, that forms acid. Okay, and that's a kind of a central thing of what I want to say here. Anyway, so the bleaching part of it is kind of well known. There are people trying to understand what the mechanism of bleaching is. Something like porites, though, the thing that we're studying, it turns out is fairly resistant to bleaching, at least for the temperature ranges that have been experienced so far. But it's been observed that it doesn't grow quite as quickly. And so that's one of the things that my collaborators really want to understand. And so the way that they try to understand this, first off, is trying to understand how it is that these things make, sort of limestone reefs, an exoskeleton. How does that happen? And once you understand that, you can then sort of ask what particular parts of that process are affected by increasing temperature or perhaps acid, that kind of thing. Okay, so this is the life cycle again. And so the things that basically, I was involved in studying are the planula and the newly settled larvae, as well as adult things. And the way you get these larvae is you have to harvest them yourself. So I had to basically, I'm scuba certified, but it turns out to do scientific diving, you have to satisfy much stricter standards. And so the University of Puerto Rico demanded that I had a real rigorous physical, and then, they checked me out and, made sure I could swim long distances and stuff like that. And even then it was kind of provisional. If you really want to be a scientific scuba diver, it can take several months to undergo the training and whatever. You have to know all of the things about underwater rescue and stuff like that. But anyway, they allowed me to do it. And so the idea is what you do is you go out early in the morning, only during a certain time of year, usually April, May, early part of June, and only in those months during the new moon. And for some reason, during the new moon, that's when these guys sort of spawn and the planula go. I guess some people think that maybe with a lack of light, the planula, are more likely to escape predation or something like that. Okay, so this shows my the main principal investigator on here. That's Loretta Robertson, and it shows her, sort of setting these traps and things. So the way these things work is you take a chunk of coral, and she basically takes a hammer, and just breaks up these porites in little chunks, put them on the ground, and then you put one of these traps over them. The trap consists of this meshwork that has very tiny, sort of like screen-like stuff, so the planula can't escape through them. But there's like a hole through the top, a tube that they can swim upward into this kind of trap. And the trap is shown here. It's kind of like a roach motel. They swim up through that narrow opening out into this thing. It's like those wasp traps. Right?
Yeah.
They can't find their way out. So it's a roach motel. They can check in, but they can't check out. Right? So each one of these things is really very, very tiny. Like I said, they're maybe half the dimensions of a grain of rice or less, and they're the free swimming guys. But what you can do is, what we did is we would harvest these things from the traps, take them back to the lab, and then once you did that, there was kind of a timing set. They would be provided with a surface, which is mostly just kind of like a bathroom tile or something like that. And these guys would swim around, and then they'd look at this tile and say, that's kind of nice. And they'd sit down and start to differentiate. When they differentiate, they look like kind of like that. Really beautiful things when they start to develop and whatever. Very elegant kind of stuff. So that's what we call a newly settled larva. All right? Okay. So the question is that we were trying to address is ultimately is how is exoskeleton or formation affected by these guys in changing temperature and things. So we haven't actually done those studies quite yet. What we did was, my role in all of this was to try to work out the methods of studying or assessing the role of several key proteins that these coral make in reef formation, all right? So again, this is sort of the coral anatomy. I just wanted to, again, remind you that, you know, in this part of the stalk, these cells have to secrete things that form the calcium carbonate. Okay, so calcium carbonate in water is insoluble. And again, they consist of two layers. We'll see, you know, several other things. But we're asking the question, basically, how normally is this actually formed by these guys, by a living organism? How do they do that? Okay, so this is a very complicated and busy slide that kind of lists all of the possible things. I'll simplify it for you in the next slide, but I just wanted to point out that the part that actually forms the calcium carbonate, allows it to form, is actually the outer layer, the ectoderm, okay? Right? In between the ectoderm and the endoderm is something called the mesoglia. It's just kind of a gelatinous kind of space. But what essentially happens is what this cell does is it, first of all, forms bicarbonate, okay? And then it transports that bicarbonate into the space where it wants to form the skeleton. This is the outside world, right around the base. Let's say this might be existing coral or a rock or something like that. It's a very narrow little space they call the calicoblastic space. So bicarbonate is put there, and then they also transport calcium there. And calcium and bicarbonate together under the right conditions will form calcium carbonate. Okay, so I thought I'd have a, you know, just, it might be fun to just do a demonstration of this kind of thing about bicarbonate stuff. So I brought in, this is a bicarbonate solution here. And most of you folks might just well have done this when you were, you know, kind of little and whatever. But if we add acid to that, that acid will combine with the bicarbonate, and that reaction will go to the left, and you'll produce CO2, right? So I'm just going to add a little bit of acid to that in the form of hydrochloric acid, all right? And you can see the bubbles forming. I put a little detergent in there, basically, to just show you the bubble formation. So I think we've all kind of done that as kids or whatever, right? So that's the common thing that you're interested in. And as I'll show you in just a bit, this is actually the reaction that occurs in the human body a lot, that our kidneys help us out with, because we produce a lot of acid in our normal metabolism, and we have to get rid of it. And so what happens is that the body, through the kidney, makes bicarbonate that neutralizes the acid and converts it to CO2. So what do you think happens to the CO2 then?
Exhale. You exhale?
You exhale it, exactly right. So that's exactly what you're doing when you're respiring. You're getting rid of the acid through it being converted to CO2, okay? That's why respiration is so important, because that's the way you get rid of your body's load of acid. And if you have compromised respiration and you can't breathe very well, what happens is your body becomes acidic. That's part of the symptoms and problems of inadequate ability to breathe. Okay? Okay, so that's fine. But on the other hand, that's not what coral does. What coral does is it basically takes that CO2, okay, that's produced in the ectodermal layer, and with an enzyme called carbonic anhydrase, it combines the CO2, and the CO2 is just, like I said, dissolves, in the water. That's what's happening in, with our increased, global warming and CO2 and everything like that. It combines in the water to become carbonic acid, which then dissociates into bicarbonate and the hydrogen, okay? So that exists in that ectodermal cell layer, and what happens is the ectodermal cell layer has, if you kind of saw it on the previous slide, it has a transporter that puts the bicarbonate out into this calicoblastic space, the space where you want to deposit the exoskeleton, okay? So what happens is it goes out there. There's also calcium that's transported from the cell layer. It's also present in seawater. It's just around. These two things combine, basically, and they form calcium carbonate, which is your exoskeleton, which is insoluble in water with a free hydrogen ion. Okay, so I can actually just sort of demonstrate that, how that works. So notice that this is clear, kind of clear fluid, and what I've got here is I've got a calcium chloride solution, and you just basically can just put a little bit in, like a little bit dark, but you see that becoming milky? Yeah, that's calcium carbonate. That's, you know, limestone, whatever, you know, when it kind of gets into things. So that's what these little beasties are doing, right? Now, the reason you have to have carbonic anhydrase is because the concentrations of CO2 are actually, you know, even though they're increasing in the world, they're actually pretty low. It's about a 1/10% or something like that of the, you know, the atmosphere is CO2. And so this reaction wouldn't run very quickly unless you had an enzyme, carbonic anhydrase, to catalyze it, to make that go quicker. So carbonic anhydrase is actually essential to this kind of thing, right? So my collaborators, what they did is they looked at what the gene for carbonic anhydrase was doing when the animal was making a transition from the larval stage, where it doesn't have to make a skeleton, to the newly settled phase. And what they found is that that gene got upregulated, activated by a factor of several thousand. Pretty amazing. That didn't tell them, though, anything about the protein. So that's where they enlisted my help, which is they wanted me to kind of develop a method for looking at these critters, you know, just actually the live critters in a microscope or something, and determining how much carbonic anhydrase there was. And then the idea would be, so you can keep these guys at different temperatures and ask, what's happening to the carbonic anhydrase? Is elevated temperatures, is it going down? Does that explain why reformation is slowed? Okay, so what I had to do basically was I found in the literature a method that's about 60 years old for actually staining tissue that has carbonic anhydrase. And the main tissue that we know, I think I mentioned, is the kidney, all right? And what the kidney does, let me just go back to this kind of thing, is what it does is it neutralizes our, I think I said, our acid with bicarbonate, and allows it to convert to CO2, which can be expelled by the lungs. The problem with this mechanism, of course, is that every time you do that, you destroy a bicarbonate. And that's the kidney's role. The kidney actually makes up for the lost bicarbonate. So it synthesizes bicarbonate, and it does that because it has, can do that quickly because it has carbonic anhydrase. So I just basically established the technique in my laboratory. This is just a bit of kidney tissue, you know, from a mouse. And the dark areas are basically these little tubules that have a lot of carbonic anhydrase. These are the areas where the kidney is synthesizing bicarbonate. I don't know how many of you know anything about kidney physiology, but there's some round little things, you know, up there. Those are the glomeruli that filter things. They don't have carbonic anhydrase. Some parts of the tubules don't, some parts do. Anyway, the whole point about doing this was that I, you know, kind of resurrected this old method that hadn't been used for years and got it to work. And then for the first time, we applied it to coral. So what I'm going to show you now are a couple of things of the newly settled larvae that are settling down and basically starting to make an exoskeleton. So this is a section through it. You take, you take this larva, which is, you know, really, really very tiny, and you embed it, and then you take a very, very fine glass knife and just cut a section of it. And then under the microscope, you can stain it and then look at things. So you can see that outer layer, that's the ectoderm. The ectoderm is dark. right? And the inner layers and things like that are much lighter. So that shows you, as I was saying, that, that in that other diagram, that the ectodermal layer is rich in carbonic anhydrase because it's trying to make or secrete that calcium carbonate to make the exoskeleton. Here's another one that's more advanced. This one you can see these things in here are the tentacles. You can actually see that the tentacles themselves, the external layer has carbonic anhydrase, but mostly it's in this basal kind of layer here. This is the part that's going to, that on this particular beast, it was removed it from where it had settled, just shaved it off and then processed it and everything. So it was basically already producing the skeleton. Here's a high power thing of it. Again, you can see this is the external layer here, right here, and this is the internal layer. You can see these things here. You can see they have a different color. Those are the zooxanthellae themselves. And here's another picture of it there. But this is from an actual adult polyp that's actually in a piece of coral that we managed to kind of get out. And this is the ectoderm, very, very dark, right? A lot of carbonic anhydrase. And these are the individual zooxanthellae themselves in the inner layer living within the cells. You can see a very interesting thing that around the zooxanthellae on the outside, there is carbonic anhydrase. So it appears that the zooxanthellae themselves also, in addition to making things like glucose, they also make some carbonic anhydrase as well to help out in this process. Okay, so that's the physiology part of it. It kind of came, back to that from all of the, the old biology, and that's why I'm involved in this thing. So the next thing up is basically, we have a whole library of specimens that have been kept at various temperatures, and to ask the question of what's the effect of this increasing temperature, we keep them at different temperatures on, first of all, how many zooxanthellae there are. We already know that with increasing temperature, they get, less dense, they leave and whatever, but this hasn't been studied. What happens to carbonic anhydrase expression? Is that going to get lighter in the things that we saw? And other proteins involved in these transporters that transport bicarbonate into that space, or calcium into that space. Okay, so I just wanted to tell you who really are the people involved here. All of these guys together, including myself when I was part of it, they called themselves Team Larva. But the people with the arrow, this is Loretta Roberson. She's the principal investigator. So she was at the University of Puerto Rico, but she's now at Woods Hole. And Rob, I think you've met Nady Bell. You might have too, Megan, as well. Nady Bell is doing her master's thesis on this topic. And so she's due to come back to the lab to do the experiments on the different temperatures and see how that affects carbonic anhydrase and whatever. But anyway, it's a lot of fun doing this stuff, although it's pretty, it's hard work, doing this stuff, diving and whatever. The conditions are pretty different than just sort of a recreational dive. There's a lot of wave action and chop, low visibility, and you have to be careful about your air supply because you have to stay down for a couple of hours and do this kind of stuff. So, but anyway, I enjoyed it.
Cool.
So, you guys have any questions about it?
Was that area affected by the hurricanes that went
yeah,
through
so
there?
I've been told that this area, being in the southwest corner, the hurricane went from the southeast at an angle across to the northwest, and it largely spared that particular town. I think that they said that things were okay. But they do get a lot of weather there anyway, so there's a lot of wave action and stuff like that on the coral.
So if we have the opposite effect of, instead of water increasing in temperature, if the water decreased in temperature, how would that affect coral?
It's a great question too. if things kind of cooled off. These things are adapted, these various corals, to very narrow temperature bands. So in fact, they're corals that go all the way up to New England and whatever. And they're adapted with different zooxanthellae that do well at lower temperatures. And my guess is that if the water became too cold, these things would die also.
How long has coral been around?
Long time. Many moons. It's first of all, it derives from a very early on in the evolutionary kind of tree. Although it's obviously evolved, but it's still relative to the complexity of higher animals and whatever fairly simple. But the other thing is it's been around a long time because there's so much limestone around. I mean, all of South Florida is limestone, basically. The whole Yucatan is limestone. There are whole areas of the world that have just been composed of just reefs that have just been deposited over the millennia, millions of years, so they're very big and extensive.
So are you going to be going back to Puerto Rico sometime again
Don't
soon?
know. I don't know. Maybe. I don't know what Loretta's sort of status is. I went there 2 years ago, and so now this coming season would be the other team. I wouldn't mind going. It's like I said, it's kind of fun, interesting. The marine station is kind of interesting too. It's just on a little island. It's just a couple hundred feet from shore, and so it's kind of isolated. Since the people on it, they're all biologists and ecologists, they have this live and let live attitude. So the place is just crawling with multiple species of these enormous iguanas. And these things harass you constantly. Couldn't believe it. Just say, come on, we've got to off a couple of these things, But anyway, it's an interesting place there. You just can't sit in anyone's space for a while because they just kind of will crowd you just begging for food and stuff.
Oh, they beg for food.
Yeah. No,
They don't want.
They
They
don't
want the
eat people
snuggles.
yet,
Like they
you know.
want. They want to be in your lap and help you research.
Well, they make good pets. If you get them young, they're reasonably well behaved. These guys know. They're always fighting with one another. There's several different species. There's one species that's it's found nowhere else in the world other than on this particular island and an island off the west coast of Puerto Rico. So they don't want you harassing these guys at all. I do.
Self-defense, right?
Yeah.
We have a few minutes yet. Rock, would you? I have to say, I'm really struck by the diversity of scientific interests that you have. Last time we did this a few years back, Rock presented on the retina in the human eye, or in the eye in general, and how the neural process of data that comes in through the eye. And then this is completely different, obviously. And I know you've also worked with squid and things like that.
Naked mole rats, that's another big project.
Maybe you could
next time I could tell you about that. That's really interesting too.
Yeah, just maybe into the last couple months we have talk about briefly some of the other areas of interest that you've explored in your career.
Yeah, it's sort of like now that it started before I retired, but I've always sort of, because my training and expertise is now sort of, I guess you could call it antediluvian. It's old. When it has uses, nobody else knows how to do it. It's mostly biochemical and stuff like that. I trained, in physics and biochemistry and whatever. And most of the current crop of folks are kind of different. So I'm sort of like a gun for hire, or not for hire because nobody pays me. It's kind of fun. I get to work on all of these very sort of strange things. So there's this. There's a thing that I'm doing in-house with Ethan Hughes that has to do with some very interesting, unique parts of the brain that have just been kind of discovered and trying to figure out what functional role they have. It has to do with myelin, things that are affected by multiple sclerosis. So his lab is interested in that. But this is in normal kind of tissue, so there's that. There's the retinal work that still do, which is sort of how the retina kind of works, but from a different point of view. We have people here, like Alon, Oleg Podolsky, he's interested in retina and how the circuits work in that, and so is Joel, I think, Silverberg as well. Let's see, and then there's another thing, which is with Loretta's husband, Josh Rosenthal. I've known him since he was a grad student way back when. He's now directing a division at Woods Hole, and that has to do with something very different. Not so very long ago, people found out that that the way that proteins got made and genes were expressed wasn't what they call the central dogma, which is you have a gene, it's transcribed into messenger RNA, and then the messenger RNA is translated into a protein. That's called the flow of information from nucleic acids, DNA to RNA to protein. It's called the central dogma. What has been found is there are now a lot of exceptions to that. And Josh works on one of them where once the messenger is made, there are enzymes which can actually change the information in that RNA so that the protein you get is different from what was originally encoded. And those are called RNA editing enzymes. And he works on that because he's a marine kind of person in invertebrates. And what he's shown, he gets to do really cool stuff, like go to Antarctica and look at squid in Antarctica versus squid in Puerto Rico or something like that, and finds that the same rough species of squid, the sequence of the proteins is different, and they're changed by these editing enzymes to take into account the fact that the temperatures are different. Squid are cold water animals, right? So if you're working in Antarctica, you're living in an environment where your body's at freezing. Whereas if you're in the Caribbean, it's probably, up in 80s, maybe even 90 degrees or something like that. So those differences in temperature make profound differences in the way proteins work and whatever if something else didn't change. And what changes is that these enzymes basically can respond to temperature changes and edit the sequence so that the protein works the same at different temperatures. So I got involved with that with him. Again, I made markers for it, and we looked in squid at Woods Hole and whatever to see where these proteins are, where they're editing. And what he finds is that squid and invertebrates without backbones use these things, these editing enzymes, very, very extensively. So it's a very interesting part of biology. Mammals use them as well, though. And so it's thought that that's going to be important. So I do that kind of work. So it's squid, the naked mole rats. Naked mole rats are I don't know if any you guys have heard of these things. I think Rob has, but I can talk some. But they're really amazing little critters that live in colonies. They're kind of like the mammalian equivalent of bee colonies. They have a queen and stuff, and they live underground in Africa. And they have a number of features that are really stunning. And one of them is that they never get cancer, and they live for 30 years. So you think about, if you've kept mice or rats, they live for a year or two. These guys live for 30 years, and they never get cancer. So there's intense interest about why that is. The other thing that's really amazing about them, which is how I got into it, is that they're totally insensitive to certain kinds of pain. They don't have pain. And it's thought because they live in these crowded burrows of 300 individuals, maybe in a small area that, that would normally be very painful. They're all breathing out carbon dioxide, so that would be painful or whatever, that they've adapted so that they're insensate to, to the pain associated with that. And I've studied that and found reasons for why that is. So that's the other. So you can see a lot of this is sort of applied by stuff. If you get a lot of experience in various things, people beat a path to your door. So it's really fun. It really is. It allows you to kind of travel.
All right. Well, thank you, Rock, very
Thank
much.
you.
Thank you.